So hello friends, so in this video I am going to discuss a question that you can see in the background, and now I am going to discuss the answer of this question with the help of the book Apurva Sastry, and also I am going to give you the reasons for choosing the right answer, that is why the chosen answer is correct and why the remaining answers are incorrect. So stay tuned. So friends, so here I have written the question. So see that which of the following statement is true about Trichomonas vaginalis. So these are the options. So it is not an STI. STI, that means sexually transmitted infections. Option B, weight saline mount of vaginal secretions host cyst. And number C, that option C, that is twisting motility seen on weight saline mount. And option D, that is it cannot be cultured. So as I have already mentioned that I am going to explain all these options and I am going to tell the correct answer, but for telling the correct answer I am going to take the help of the book Apurva Sastry. So let's see. So see there, so this is a Trichomonas vaginalis. So see that, so Trichomonas vaginalis is a flagellated protozoan of the genital tract and causes trichomoniasis. So trichomoniasis is caused by Trichomonas vaginalis and it is the most common parasitic sexually transmitted infection. So you should remember that it is the most common parasitic sexually transmitted infection. So now, so by seeing this you can eliminate the option A. Why? See. So here that is option A. So here says that it is not an STI, that is it is not sexually transmitted infection. So but here it says that, so here it is trichomoniasis is caused by Trichomonas vaginalis, and this Trichomonas vaginalis, it is the most common parasitic sexually transmitted infections. So therefore that is I can say that Trichomonas vaginalis, so here causes trichomoniasis and it is the most common parasitic, so here sexually transmitted infection. So therefore option A is wrong. So you can that is eliminate the, so here that is option A. Now, so here now let's see that is morphology. So that is it has only a, that is trophozoite stage. There is no cyst stage, and trophozoite has two forms. So here flagellated and amoeboid form. So now here also it gives clue that about the option B. So here that is option B says that is a weight saline mount of vaginal secretion so cyst, but till now we have read that there is no cyst stage. Now, so here that is, let's see the life cycle. So that is a trophozoites are ineffective form, but so here transferred by sexual route they transform into amoeboid form. So here that is in the human being. So here that is trophozoite. So here cause infection, genital infections in this amoeboid form, not in this. So here that is a flagellated forms. Now, so here that is they again transform back to flagellated trophozoites that are discharged in vaginal or urethral secretions. Now, so these are the clinical features. So clinical features you can see that. So here that is the discharge, vaginal discharge is found, and in females that it causes vulvovaginitis. Now here that is strawberry appearance of vaginal mucosa, that is colitis mucolaris is observed. And also that is other features, that is dysuria and lower abdominal pain. And number 4, that is in males it presents as non-gonadal urethritis. So these are the clinical features, that is strawberry appearance of vaginal mucosa is observed in 2% of patients, and why? Due to punctured hemorrhage spots on vaginal and cervical mucosa. So now let's come about a laboratory diagnosis. So here is a laboratory diagnosis. So here it is the weight saline mount of fetch vaginal discharge samples can demonstrate the jerky, motile trophozoites and pus cells. C, that is the weight saline mount of fresh vaginal discharge or secretion samples can demonstrate the jerky, motile trophozoites and pus cells. So here in this, so you can eliminate option B also. Why? Because option B has said that it is the weight saline mount of vaginal secretion so cyst, but here it is, so here in this case it is wrong. Why wrong? Because there is a weight saline mount of fresh vaginal discharge or samples don't show the cyst, they demonstrate the jerky motile trophozoites and pus cells. Now, and also that is about the morphology. You can see that not about the morphology, about the motility. You can see that is a trophozoite. It is a pear shape and measure 7 to 23 micrometer, and this trophozoites that is shows that is characteristic jerky and twisting motility in saline mount preparation. So in the saline mount preparation, so here it shows that is twisting motility, jerky or twisting motility. So therefore you can see that here you can also see that the option C, that is a twisting motility seen on that is with saline mount. Yes, so here it says that the trophozoite shows characteristic jerky or twisting motility in saline mount preparation. So now here you can say that the option C is correct. Why? Because it is saying that twisting motility seen on that is with saline mount. See, now here I explain both these options B and C together. See, that is the wet saline mount of vaginal secretions can demonstrate the jerky motile trophozoites or twisting motility of trophozoites and pus cells. So here again that is the twisting motility seen on wet saline mount. So therefore that is option C seems correct. And also you can see that the option B about written, that is a wet saline mount of vaginal secretion. So here that is don't show cyst and no cyst stage is found. So here also but option B is stating that is a wet saline mount of vaginal secretions for cyst. So therefore that is I can eliminate that is option B, that is option B is wrong, and this shows that option C is correct. Now let's discuss about does the elimination of that is option D. That is option D says that it cannot be cultured. It cannot be cultured. That is a Trichomonas vaginalis cannot be cultured. So let's see. So this is a laboratory diagnosis. So the first laboratory diagnosis is that the wet saline mount of fresh vaginal discharge, and as I've already understood that on the wet saline mount of vaginal discharge, that is the jerky motile trophozoites or a twisting motility of the trophozoites seen in saline mount preparation. Now so this is for a diagnosis, and also that is morphology. About the morphology given, that is the 5 flagella, that is a 4 entire flagella and 1 recurrent flagellum, that is the and also this a single nucleus exotel. So this is the morphology that you will see in that is the microscopy. And other, so here that is diagnostic modalities for trichomoniasis include that is a culture. So here that is a culture such as that is the lace cystine, that is hydroxylate serum medium. So if you say yes, it is cultured by using a medium that is lace cystine hydrolysate serum medium. So here also you can see that this also eliminates option D, because option D says that the Trichomonas vaginalis cannot be cultured, but it is false. So therefore that is I have written that. So here there is a Trichomonas vaginalis. It is cultured by using medium such as lace cystine hydrolysate serum medium. So therefore option D is wrong. So once again, so why option A is wrong? Because Trichomonas vaginalis is the most common parasitic sexually transmitted infection. Option A is wrong. And wet saline mount of vaginal secretions can demonstrate the jerky motile trophozoites or twisting motility of trophozoites and pus cells. And wet saline mount of vaginal secretions don't show cyst and no cystine. So therefore option B is wrong and option C is correct. And also that is Trichomonas vaginalis cultured by using media such as lace cystine hydrolysate serum medium. So option B is wrong. So that's all.